Hi everyone, welcome to Basic Science Series. I'm your host Ravinder Kumar, and today we will understand the microbial world, we will understand the members, diversity, and the classification system. This is an introductory video where I'll show you why microbes are diversified, how we study microbes, what is microbiology, and what is the classification system of the microbes. We'll not go deep into the classification system, but I'll give you some of the important and basics of the classification system, and I also, as you can see on the slide, I'll also explain why we have different groups and how they are further divided into three major groups, which are Archea, Bacteria, and Eukarya. So let's start the video and try to understand what microbial world is, how we classify it. Whenever we talk about microbes, whenever we talk about microbial world, we have a lot of things to discuss. There are numerous microorganisms, diversified microorganisms present around us, present in areas where even we cannot imagine. So microbial diversity is basically huge. And now in this case, before we study the classification or discuss about the classification, we need to understand what is microbiology. Microbiology is nothing but the study of microbes. When we study microbes, when we study their different features, when we study metabolism, when we study their structure, all is included in the microbiology. And what are microbes? So microbes are the organisms. They are too small organisms, too small to be seen by unaided eye or naked eye. So they are very, very small. We need sophisticated instruments to study them. The instrument that we use to study microbes is a microscope. A microscope is nothing but the optical instrument to study these tiny organisms called microbes. And since we need to classify them, so we need to find out specific features. So initially, when we developed the microscopes, we were able to identify what are the different features. I hope you remember who discovered who invented the microscope. This is one of the important questions in microbiology. Now, when we want to classify them, and we need distinct features. Now, before we discuss those features, as you can see, I have written some of the organisms, even microorganisms, they can be observed without microscope. Examples are filamentous algae, bread mold, and two bacteria, which are Thiomargarita and Apulopicium. These are the two organisms. Bacteria specifically, you can observe them without the use of microscopes. Now, when we talk about their classification, two things, they're important. One, high complexity and diversity. So you have highly complex structures, and they are really hard to study because they are tiny. Second is the diversity. They are diversified. So it is very, very hard to classify them. Initially, what we have done is, based on one prominent structure, which is nucleus, we divided them into two different groups. One is prokaryotic and second one is eukaryotic. So based on the presence or the absence of the structure known as nucleus was one of the prominent structure. Essentially, I can say is one of the prominent structure that is present in the cells. So it was easier to distinguish them into two different groups. Nucleus is nothing but the organelle that contains the genetic material. So if the membrane-bound organelle is present, that means eukaryotic organisms. If it is absent, that is prokaryotic. Now, another feature, in case of prokaryotic cells, they were smaller, they were simpler. In case of eukaryotic, they were complex and they were large. So that is another feature. So this is how you can see simple features you can use to classify different structures, different cells. But then we wanted them to classify because there were so many features, and discussion of those features will be beyond the scope of this video. But then slowly, based on their features, we have classified them into Monera, Protista, fungi, Animalia, Plantae. These are important 5-kingdom classification system. So here, in case of Monera, we have bacteria and Archea. So this is what we have classified. Later in 1970s, Carl Woese, he used or we can say he suggested the use of rRNA sequencing techniques. Based on that, surprisingly, we found a lot of conserved regions, and then we were able to classify these organisms based on the specific rRNA sequence. And then Archea, Bacteria, Eukarya were the three dominant groups that emerged, and based on specific features, further they were classified. It is essentially on the basis of the rRNA sequence. So then they were connected. Obviously, many of the organisms, they emerged from one another, and then there was one common ancestor from where these three groups were evolved. So you can say it's a process evolution where you have different organisms coming from a common parent, and then complexity and diversity increases. So this is all about the microbial world. We have discussed the diversity, members, classification. As you can see, in this one, we have just discussed the name of the five kingdom classification system, and then we went into the rRNA sequencing. We found out Archea, Bacteria, Eukarya based on this particular sequence. And then slowly you know that the further complexity is going to increase, especially in case of all these three important groups. So I hope now you understand a little bit about the classification system. I want you to study more on this particular topic, because this is just a pointer. It is going to give you some hints where you need to study more. For example, you need to study the features of Archea, Bacteria, and Eukarya, and with that you will be understand why they are classified into a specific group. So I hope that this video was helpful for you to understand the microbial world, and I'll bring more videos, and if you want to watch those videos, then please stay tuned, support the channel. Thank you and take care.